Hey guys, in this video we're going to look at a little bit about DNA. Right, this is an introductory video to this unit. So let me get this thing pulled up here. There we go. All right, so we're going to talk about the discovery of DNA. Just briefly, a little bit of history. We're going to look at its structure, and we're going to introduce how DNA replicates itself. There are some pretty important questions that we want to be able to answer by the time that we're done with this. And so keep these in mind as we go through. We want to be able to describe Rosalind Franklin, James Watson, and Francis Crick's job as far as how they determine the structure of DNA. What did they contribute? We want to know the three parts of a nucleotide. We want to know base pairing and what base pairs with which. And we want to obviously be able to describe how DNA replicates itself. So let's start with something simple. What is DNA? Well, DNA stands for deoxyribonucleic acid. So let me get my pen started up here. There we go. The DNA, deoxyribonucleic acid. Now, ribo comes from ribose, which is a sugar. And hopefully you will remember that because one of the parts of DNA is a sugar, all right, the ribose. Deoxy obviously means something about missing oxygen or an oxygen that's been removed. And then the nucleic acid, this tells you, hey, this thing is acidic, okay? It is an acid, all right? So a lot of information is packed into that word. Now, a little bit of history for you. So in 1928, a guy by the name of Griffith established that something was transferring traits from, parent to son, parent to daughter, whatever. We know from Gregor Mendel that it had already been established that genes, right, were the vehicle of those traits, right? That's how the traits were transferred. But they didn't actually know what the molecule was. In 1944, all right, so right at the end of World War II, Avery identifies DNA as what he called the transforming factor. He said, hey, DNA is what codes for this information. And in 1952, these two guys, Hershey and Chase, they tagged DNA with a radioactive isotope, which allowed them to track it from cell to cell, and they confirmed his findings. So for about 25, 30 years there, scientists are looking closely at this DNA molecule. Now, in the 50s, Rosalind Franklin got this pattern right here, this X pattern, see it right there, in an X-ray crystallography experiment. So one of the ways that we try to figure out the shape of a protein is we will encase it in a crystal that keeps it nice and straight. So we have our protein right there, and then we put it in a crystal, and we X-ray that crystal. All right, so that's called X-ray crystallography. And Rosalind is pretty important historically. I mean, she's one of the first women to get credit in a major scientific discovery. She's not the first, but she's one of the first. The same time this is going on, where she's trying to figure out what the shape of this molecule is, there's Char is working out how the bases in DNA are pairing. So they knew that there was adenine, thymine, cytosine, and guanine. Those are the names of the bases. And they were trying to figure out how they corresponded to each other. How did they, work together to build that DNA ladder? And the last thing that happens in the early 50s there that's of huge importance is the actual double helix model is submitted by Watson and Crick. And so Watson and Crick get credit for saying, hey, DNA is double helix, all right? It's a twisted ladder. So a lot of times when we just sketch it, we want to sketch it like this. So we'll draw two intertwining kind of like shoelaces, and then we just kind of alternate our ladder so it looks like it's been twisted. So this is the shape that they came up with. And we've already talked a lot about chromosomes, okay? We talked about chromosomes when we did mitosis and meiosis. And, we just kind of say, hey, there are these chromosomes, and on the chromosomes are genes, but we didn't really go into depth about what is DNA and how it's related to a chromosome. So this picture right here is key for you to understand how does DNA become a chromosome. So DNA is already coiled, right? We know it's twisted, right? We got that nice little ladder thing going on there, all right? But when you twist the twist, it kind of coils up right here. If you've seen one of those old corded phones, it kind of looks like that. And the more you twist it, the tighter it gets. And so a chromosome is actually what we call condensed DNA. So this is a condensed version of DNA. And the thing about a chromosome is you only see a chromosome when a cell is splitting, because you can't read the DNA when it's all wrapped up like that. So you're only going to see a chromosome when we're talking about mitosis or meiosis. You shouldn't see a chromosome in a cell when it's undergoing interphase. Now, you probably know from, again, when we talked about meiosis and mitosis, a lot of these things end up going together. That you, as human beings, we have 2 copies of each chromosome. Each pair have the same loci, but not necessarily the same copy of the gene. All right, so that's new. So each one of these little bars right here in this picture is called a locus. And that is where you find a gene, a specific gene. So, we'll just make some up. This is the gene for height. And this is the same gene for height. But you might have H1 on this side, and you might have H3 on that side. They don't have to be the same allele. They can just be, but they do have to be the same gene, right? So they're copies of each other, those 2 chromatids, those sister chromatids, right? But each location on the chromosome that has a corresponding gene is called a locus, and the plural is loci. When we're talking about those genes, all right, a gene, the definition of a gene that we want to use is that a gene is a unit of DNA that codes for a specific protein or a polypeptide. Remember, proteins have peptide bonds, and poly means many. So many peptide bonds, many proteins, that would be a polypeptide. Now, there's a lot of different parts to a gene. There are promoter regions. There are open reading frames, which is one that we definitely will talk about, which is actually what gets coded for our protein. There's a terminator sequence, which says, hey, stop reading the gene. You've made the protein. We need to cut it off. And then there are enhancers, which are areas that, if they're turned on, will make your cells make more of that protein. If they're turned off, obviously it will suppress that protein. Now, we're in introductory bio, right? We're, 9th, 10th, maybe you're an 11th grader in bio, but this is bio 1. So we're not going to get into a great amount of detail with this. The important thing that I want you to take out of this slide is genes are not just simple little, strings of letters. There are different regions that do different parts, that do different jobs, excuse me. All right, so here's Crick's sketch of the DNA molecule. It is 2 chains, so think about it as chain 1 with 1/2 of the ladder, and then chain 2 with the corresponding half of the ladder, and we put those together, and they twist, right, and we get DNA. Okay? When we say double helix, that's what we're referring to. The 2 different strands. So here's in his drawing, strand 1. All right, and then here, we'll do a squiggly line, is strand 2. Okay? We usually describe it as a twisted ladder. This is really just more of a descriptive thing. The individual nucleotide, so if we zoom in on the DNA ladder itself. So DNA, right, we'll draw it again, and you should probably get used to drawing this because it helps. So there's the ladder. We know it is broken down into 2 strands, right, just like that. And now we're just going to look at one of those strands, one of the nucleotides. All right, so each little ladder, each little rung in the ladder is a nucleotide. It's got 3 parts. It's got a phosphate backbone. Right, this is the backbone right here. And that phosphate, let me get to green. match. Oh my goodness. Right. So the phosphate backbone is that line right there. That's the backbone. Okay. And then the sugar and the base form the rung, the step ladder part, right? So the sugar and the base are right here. That's the sugar and the base. So there's the nucleotide. So three main parts. The backbone is phosphate. Sugar is our linkage over to the base. And this is a nitrogenous base, meaning it has nitrogen. All right, that's what we mean when we say nitrogenous base. And you can see nitrogen, 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 all over the place, nitrogen, right? There's a bunch of nitrogen in our nitrogenous bases. There's four bases in DNA: adenine, thymine, cytosine, and guanine. We abbreviate them A, T, C, and G, okay? The purines are A and G, and the pyrimidines are T, C, and U. U only shows up in RNA. All right, what's RNA? Well, DNA is this nice twisted ladder, right? That's DNA. There's also RNA in your body. We use it for different things. There's rRNA and mRNA, tRNA, which we'll talk about all through this thing. But RNA is always a single strand, and there's no T in RNA. All right. Where'd my pen go here? Come on. There we go. Why is my pen not working? There we go. Okay. So RNA has a uracil. A always bonds with T. C always bonds to G. A will also bond with U because U and T are interchangeable. There is no T in RNA. There's a U there. And that'll be important later. So what do we mean by this bonding? What does that look like? All right. Well, you can see these little red dashed lines are all hydrogen bonds. Okay? Hydrogen bonds. Now, we talked about hydrogen bonds back when we did water chemistry, and we know that hydrogen bonds are not real bonds. They're just forces of attraction. It's the same hydrogen bond that like holds water together, gives a good surface tension and cohesion and adhesion and all those things. And the reason that we need those hydrogen bonds and not real bonds is because at some point, DNA has to unzip. It has to break apart so we can actually read which bases are on each side. And if they were totally bonded, well, you couldn't break it apart. So it's really important that there are hydrogen bonds in place there. Now, A and T make two H bonds, right? 1, 2. C to G makes three H bonds. 1, 2, 3. There we go. All right. All of the base pairing happens by hydrogen bonds. We just said that. We call those bases that hydrogen bond with each other complementary bases. Okay. You can kind of think about this in terms of like holding hands, right? They need to match. So because we have 5 fingers, we can go like this. Imagine if you had like 9 fingers on one hand and you try to hold hands with a 5-finger print. Well, it would kind of work, but it wouldn't be as stable. You have the same issue with A and T compared to C and G. C and G want to make those nice 3 hydrogen bonds. A and T want to make 2. So you can force them together. That happens when we talk about mutations, and we'll look at that. But the most stable arrangement is going to be C to G and A to T. Now, this is called Chargraph's rule. A goes to T, C goes to G. And Chargraph's rule is pretty simple, all right? I want you to be able to identify Chargraph is the guy who said, hey, look, we got to get A bonded to T, C bonded to G, okay? That's all you need to know about Chargraph's rule. I mean, that's really all that there is to Chargraph's rule, okay? So make sure you know that, all right? So let's look at the structure of DNA now. All right, we're switching gears a little bit. DNA structure is comprised of nucleotides in long chains. So we call them polymers. Poly means many, mer means part, so many parts. We describe the chain just by listing the sequence of bases. So we say, like, this chain is ATGACG. That's it. Since we know Char's rules, since we have base pairing, we know the other strand would be complementary. So what do I mean by that? Well, let me move my face over here out of the way. So it's complementary in that in this previous slide, we have ATG, right? That's what we start with. We still have A, T, and G, but I know what's on the other side because if I see an A on strand 1, and on strand 2, I have to have a T. If I see a T on strand 1, must be an A on strand 2. G must be hooked to C, C has to be hooked to G, so on and so forth, all right? So that's why we need to know Char's rule. That lets us predict our sequences, okay? Now, when you see this in a double helix, all right, you can still do the same thing. Just make sure you know that these are straight across, all right? There's not anything weird going on. Now, there's a pretty cool animation on YouTube that goes into the molecules, all right, that are involved in this. And this is what DNA replication really looks like. You have all of these different colored molecules, and they're unzipping the DNA, reading one side, and then putting together another side so that they can replicate that DNA. Obviously, this is really, really complicated when it comes down to identifying each molecular machine. We don't have to do that. I do want you to watch this video. Turn the sound on. It describes a lot of stuff. It's interesting. It gives you an idea of what we're about to talk about in real time, but we're going to look at DNA replication just as a stepwise process. So let's look at that. So the first step of DNA replication, you have to break down your hydrogen bonds. You have to get the DNA to come or to be able to come apart. And the second step is then going to be an unzipping of the DNA. Now, in this picture that I've drawn here, it almost looks like the tail end of the DNA is, being just left to dangle. That's not always true. A lot of times, the sequence that you want to read is in the middle of the DNA, and that's okay, all right? There are specific enzymes that can unzip just a portion of the DNA, all right, in order to get to there. So either one of these could be accurate. Now, once you have unzipped the DNA and those loose ends are just kind of hanging there, then free nucleotides in the nucleus can kind of just fall into place. They want to have those stable hydrogen bonds, and so that's what they do. There's free nucleotides there, they fall into that unzipped DNA, and they make our new strand. Once you've got that in place, hey, look, we've got 2 new strands of DNA. This would be something that would happen in like S phase of interphase, right? We need to duplicate our DNA to get ready for mitosis, and so now we've got it. Now, both strands are identical. You have a perfect copy. Half of each strand is old, half is brand new, right? The free nucleotides make a brand new set of, or a brand new side of the DNA. We call this old and new thing, this combination of an old strand and a new strand, semi-conservative replication. All right, why is it semi-conservative? Well, the old strand is kind of preserved, right? It's saved, but half of the strand is brand new. That new half can lead to changes, and we'll talk about those changes later on if there's a mistake in how those free nucleotides come together. But as far as DNA replication, pretty simple. Just the enzymes unzip the DNA, free nucleotides fall into place. That gives us our new 2 strands, right? It's pretty simple as far as how that works. Just make sure you know this vocab word, semi-conservative, and that you can describe these 3 steps. So the enzymes break the hydrogen bonds, the strands unzip, the free nucleotides fall in, and then we have our 2 new strands, okay? So that's what we want to get out of DNA replication. So hopefully now you can answer some of these questions. I would recommend that you do that. I also included a couple sample test questions like that you might see, just so you have an idea of what's going on. So make sure you can go through and answer these. All right, I'm not going to answer them for you, but if you're reading the PowerPoints, you'll probably notice that the answers are in the PowerPoint, right? So make sure that you have a chance to look through these. You will probably see these tests later, or these test questions, sorry, later on, okay? So make sure you have a look at those. I'll see you in the next video. Let me know if you have any questions. I'll be glad to give you a hand.